with us. I'm a big fan of your work, and each time I'm seeing one of your papers or tweets, I'm like, oh, yes, yeah, amen. So I'm really happy to have you here. I really look forward to your talk. As I said, it's like you have roughly 40 minutes, 1 hour to give the talk, and then we have like some informal discussion. I'm going to record the talk, but not the Q&A. So maybe if you want to start recording already, and Earl, you have the floor. Thank you so much.
Okay. Thank you very much, Anna. Thanks very much for the invitation. It's a great pleasure for me to be here and share my work with you. So let's just jump right in. Our laboratory studies the executive brain functions. These are the brain functions that are involved in planning, focusing attention, learning and following rules, organizing thoughts, the kind of things we think of when we think of thinking. And we do so by recording electrical signals from the brains, mainly of non-human primates. Often we do work in humans as well, human primates. And we use computational modeling and analytics to make sense of these signals. So we not only collect the neurophysiological data, we also model it and use modeling to generate new hypotheses that we can then test in the data. We have that kind of iterative interaction between modeling and experimentation. And we do so because we want to understand cognition. I mean, understanding cognition in the brain is a good thing, but importantly, we want to understand its dysfunction in diseases such as autism, schizophrenia, attention deficit disorder. So much of our work uses newish technology. And I say newish technology because probably to young students today, it's not that new. But when I started out in this field in the 1980s, we were limited to recording from one neuron at a time using a single electrode. And much of the focus back in the early stages of modern neuroscience in the late 20th century really was focused on the brain's individual parts. It was a very piecemeal, very part by part understanding of the brain. But now we have the ability to focus on hundreds or millions of neurons simultaneously, and our knowledge of how the brain works. I mean, whenever we're figuring out something as complex as the brain, we need to start by figuring out what the parts do. But now in the 21st century, we sort of move beyond that initial stage of modern neuroscience, and now we're more in the integrative mode of neuroscience, where we are trying to figure out how all the parts work together. And this allows, this new technology and this new understanding allows understanding of the brain, not as individual parts, but on the network level. It's emergent properties. These are properties and mechanisms that emerge only when the parts interact as a wider whole. And I'll get to that in a moment. But we study two types of signals with our microelectrodes. One is the spiking activity, which is the impulses from single neurons. Think of them as individual voices from a very large crowd. And we also look at local field potentials. This is the average activity of millions of neurons near each recording electrode, so the roar of the local crowd. And I'll also briefly mention we can even look above the level of local field potentials to the electric fields created by local field potentials. I'm not talking about electric fields 5 feet outside the brain or even 5 inches outside the brain. I'm talking about the electrical fields that are hovering just above the brain, just a slight level of summation above the local field potentials. But we think a lot of brain functioning happens not at the level, a lot of single neurons and spiking is very, very, very important. Everything starts with that. We think a lot of functioning in the brain is happening at this higher level of electric fields and local field potentials that can influence one another. So our main interest, since we're interested in executive brain functions, our main interest has been in the prefrontal cortex. The prefrontal cortex is critical for planning, monitoring, and executing complex goal-directed behavior. We've known this since the days of Phineas Gage, a famous patient who had his frontal lobe damaged by an accident, that the prefrontal cortex is important for all these things that make you goal-directed and act appropriately in society. Now, originally, the physiology of the prefrontal cortex mainly focused on working memory. And I'll talk about working memory a little bit, but at the time, that was the prefrontal cortex was thought to play a major role in working memory and holding things in mind, in consciousness. And that is an important function of the prefrontal cortex, but it's not the only function of the prefrontal cortex. What the prefrontal cortex does, it has much larger role in brain function. And our main hypothesis is that the prefrontal cortex must learn and use the rules of the game to understand what goals are available, to understand how to reach those goals, to understand how to behave in a society, how to behave when you're listening to a Zoom talk. To understand all that, your brain has to learn the rules of the game. What makes these rules? What are the goals and what are the ways of navigating our world? And this is what we think the prefrontal cortex must do, must primarily do that. And this requires acquiring top-down knowledge. Now, top-down knowledge is knowledge of how the world works. Bottom-up information is sensory inputs flooding into your brain. Top-down information is the knowledge, interpretations, and expectations your brain has learned from experience. And we use that top-down knowledge to predict possible goals and how to achieve them. Now, about 25 years ago or so, we started to test this hypothesis by studying the role of the mechanisms by which the brain, by which the prefrontal cortex, might possibly absorb this top-down knowledge of how the world works. Now, at the time, again, the focus was on working memory. We started a series of investigations where we began to look at how the prefrontal cortex really does acquire these rules of the game. And what we found across a series of studies supported our hypothesis that the prefrontal cortex, its neural activity is tuned by this top-down information that humans and animals learn. So one thing we did is we started a series of studies where we taught the animals categories. And one series of investigations, we taught the animals the categories cats versus dogs. Now, in this experiment, there was computer-generated images of cats and dogs. There was 3 prototype cats, 3 prototype dogs. You see the prototypes here. And what the arrows indicate are morphing or smoothing between the different prototypes. So along these different morph lines, there were smooth transitions from this dog to this cat, or that cat to that dog. But what we did is we trained the animal that there was a sharp boundary in the middle. In this case here at the 50-50 line between cat and dogs. And we taught the animal that everything on one side of this boundary was by definition a cat, anything on the other side of the boundary was by definition a dog. And you could draw this line literally anywhere and you still get the same result. But what we did is we taught the animals that there was a smoothly varying experience, but we taught the animals there was a sharp boundary, there was a sharp boundary that didn't actually exist in the real world. And what we found a whole bunch of neurons in the prefrontal cortex that reacted to the category, whether it was a cat or a dog, but were completely insensitive, did not convey any information about the individual cat and dog members. After this training, the prefrontal cortex became tuned to these 2 categories, cat versus dog, and it started ignoring all the details of the individuals, which wasn't really relevant to solve the task. Having done that, we then trained, when Andreas Nieder was in the laboratory, he trained monkeys to recognize the numbers 1 through 5. And much like the cat and dog story, we taught the animals to recognize the numbers 1 through 5 by the number of items on the screen, and these could be displayed in a wide variety of ways. The exact details of the display were irrelevant. What matters was what the numbers were, and the neurons in the prefrontal cortex became tuned to number. We then took this to an even higher level, and we trained animals on abstract rules and concepts like same versus different and up versus down. And once again, we found neurons in the prefrontal cortex that became tuned to these high-level concepts and threw away the details of sensory experience. What we found over all these studies is that the prefrontal cortex absorbs top-down information, and its neurons throw away the unnecessary details, the details of what the individual displays, of which individual cat. They throw away those details. That's something that your sensory cortex can do. The prefrontal cortex was more concerned with solving the task, so it just learned these rules and principles. And we went on to show how the prefrontal cortex could then use this top-down information to control the flow of activity across cortex. Now, this is a study we published 22 years ago. I'll go into a little more detail later. I won't go into detail now, but I will say this is pretty much sums it up, is the idea is the prefrontal cortex uses this top-down information. It's like an orchestra conductor that learns the score, learns all the information the animal needs to solve a task, and it sends top-down signals back to the rest of the cortex that acts like an orchestra conductor and tells the orchestra what to do at the right time in order to engage in this appropriate goal-directed behavior. Now, In doing these studies, we found an unusual property that we did not know about back when I was a graduate student. It's a property called mixed selectivity neurons. Now, to contrast what mixed selectivity is, consider how we thought of the neurons in the 20th century. We thought of the brain in the 20th century as being like almost like clockwork. We thought of the brain as individual parts, and if you could figure out what each part does, each neuron does, and we could figure out how all the neurons are connected, we could figure out the whole brain one piece at a time. We now know that's not true. There's all these additional properties you can't tell. You can't study one piece at a time. But back then, in this more simplistic view of the brain, we thought that neurons acted like they had certain properties, simple properties, like they detected single features like edges, or some simple linear combinations of a few related features. So under this traditional selectivity, you might find neurons back in visual cortex, a neuron that responds to the combination of red, rolling, and ball. And it's involved in this one perception. Now, I'm not here to say that we were not as smart in the 20th century or we were looking at it the wrong way. This was a necessary step to get to where we are now. We needed to figure out the brain. Back, it was only 1962 when Hubel and Wiesel first really figured out how to get information processing in single neurons. So all this was a necessary first step to get to where we are now. But in conducting these studies, we found these unusual property of neurons called mixed selectivity neurons, and they didn't fit the traditional mold of neurons detecting simple features or a few simple related features. Rather, mixed selectivity neurons are neurons that are selective for, they're activated by complex nonlinear combinations of many features with no obvious relation. And I'll get to an example of this in a few minutes. But the idea is these neurons, they didn't just do one thing. They did many different things. They could participate in many different functions, many different ensembles, because they seem to be sensitive to a variety of features that had no obvious relationship to one another. And we found that these neurons are highly multifunctional. If you train an animal to do 2 or 3 tasks, you find the same neuron will have different functions on each of the different tasks, as if the neuron is participating in a different network for the different tasks. So the neurons are not single function like we used to think about the brain, they're multifunctional. They do a variety of different things. So why are these mixed selectivity neurons so important? Why did we find them when we first started training the animals on these complex tasks? Well, back our first report of multifunctional neurons was back around the turn of the century. But more recently, my colleague Stefano Fusi Matti Regata, working with my former graduate student, now Professor Melissa Warden, they showed computationally why the brain needs mixed selectivity. And what mixed selectivity does, it adds extra horsepower to the brain, extra processing power. It's like these neurons with the wide variety of properties that can be activated by a variety of different information. They're like a neural bazaar where a wide range of information can be combined in the same network of neurons. Most of what we do every day is arbitrary rules. Our brains didn't evolve to know that red means stop and green means go. This is something we just made up. There's got to be somewhere in the brain where information can be just pulled together really rapidly and make new rules. And it seems to be this population of mixed selectivity neurons. It also makes the brain smarter. The mixed selectivity neurons provide a high dimensional, their multifunctionality, their ability to respond to different things creates a high dimensional representational space that allows the brain to solve more complex problems than it could without the mixed selectivity. And it also, for reasons, math reasons I won't get into, it also increases the storage capacity of the brain. Neural activity is highly correlated. Your brain oscillates and many neurons are all doing the same thing at the same time. They synchronize their activity. That limits the storage capacity of neurons in your brain, because if you had a million neurons and all 1 million neurons did the exact same thing, it's kind of like having just one neuron. And what Stefano, Matti, and Melissa found was that mixed selectivity allows the information to be distributed widely across a lot of neurons, rather than just be limited in just a few neurons, and that makes the brain essentially capacity unlimited. And for all these reasons, it allows the brain to learn things really quickly, and it allows flexibility because all this information is there at the fingertips of the brain, ready to be used in any way the brain wants. Okay, so mixed selectivity, this idea of mixed selectivity requires a new way to think about the brain. Again, we used to think that every neuron had one function, and we used to think that anatomy was destiny. Neurons combine signals because they're wired together. If two neurons have a connection and that synaptic weight strength is strong, when one neuron fires, the other will fire. That was how we thought about the brain. It was all about what individual neurons do and how they're wired together. And that's illustrated here. Here's what thought to be, we used to think that every neuron had that every idea, perception, action has a unique anatomical network called an ensemble. So I've drawn that here on the left here. Here's one, we'll call this one thought. This is the blue network. The idea is that this network of neurons forms an ensemble that's responsible for representing that one thought. And if you have another thought here represented by the red network, that's a completely different ensemble, a completely different network. Now you could form a few associations between these ensembles if you learn that blue and red go together, but generally each unique thought, unique piece of information in the brain was thought to have a unique network, unique ensemble. But mixed selectivity means that neurons aren't members of just one ensemble. It means they're members of multiple ensembles, multiple networks. So here we see, here's my two networks now. Here's my red and blue network, all combined, sharing common elements, which mixed selectivity implies. Now we have a red and blue network sharing common elements, these mixed selectivity or multifunctional neurons, shown in this checkered. So the idea is these neurons can participate in both the red ensemble and the blue ensemble. And this is just two networks. You know, in reality, it's probably dozens, if not hundreds, of networks overlapping one another in your cortex. So if anatomy is destiny, how does this work? If I want to activate just one of these ensembles, think the red thought or think the blue thought, how do I activate one without activating the other? Why don't all the networks activate one another and create a jumble of thoughts? Like if I want to think the red thought, I might start activating the red neurons here, the red neurons. Then that activity would run through the mixed selectivity neurons to the blue network, and now we have two networks activated, two thoughts activated, and now your brain is all a jumble of confusion. So if anatomy is destiny, how does this work? How do we actually form ensembles in the brain that are unique for each thought with all this overlapping anatomy? Well, this brings us to the topic of emergent properties. Emergent properties are not explainable by individual part properties alone. They emerge as a result of the interaction between the parts. So for example, here is a crowd doing the wave. And you can see that there's this crowd at a sports stadium. You can see the wave is moving from right to left in the slide here. This is an emergent property. No one's instructing these people what to do by having a few simple rules, like stand up when the person on the left of me stands up and sit back down again. You can create this organization among a bunch of non-interconnected and bunch of individuals. And importantly, even if you study this entire crowd, if you study this crowd the way we used to study the brain, one neuron at a time, one person at a time, you could study the entire crowd and you would never see this wave. It's only by understanding how they interact in real time that you can actually see this structure. Now, what does that have to do with the brain? Well, your brain oscillates. Your brain is constantly oscillating. Neural oscillations are an emergent property. They reflect the organization of millions of neurons. So here's some EEG traces for your brain is oscillating. These are recorded from the scalp, of course, or EEG recordings. Your brain oscillates any time from 1 time a second up to 100 times a second or more. And there's all these frequency bands that we arbitrarily divided for different frequencies. Now, what these neural oscillations represent are the organization of millions of neurons. Oscillations are millions of neurons activating together and deactivating together in synchrony. So that's organization. That's just the fact that the brain is oscillating means there's already some organization happening in your brain, some self-emerging organization. Now, if a stadium crowd can self-organize just a few simple rules, imagine what your brain could achieve with this principle. And in fact, this kind of large-scale organization is needed for top-down executive control. Goal-directed behavior requires synchronizing and coordinating large numbers of neurons toward a specific function. When you make a decision, when you pay attention, when you engage in some action, you're getting millions of neurons all synchronizing their activity to achieve the same goal, to achieve the same function. So this kind of these oscillations represent the kind of organization the brain needs to do anything complex. Now, I can't do experiments on this, but my philosophy, if you will, is what probably happened is any electrical system or neural network will naturally begin oscillating if you connect the elements together. Radios do that if you start building radios. I think it's probably likely that nervous systems got complex enough to naturally begin oscillating. And evolution came along and said, oh, I can use that. I can push around these oscillations and actually use this self-generated organization and get control over it. And now I have control over how the brain works. But also, there's something called representational drift. Neurons are fickle. If you record from a single neuron during any sort of behavior, even a simple thing like having the animal view a picture or a stimulus on the screen or a sound, what you'll find is that neurons will respond sometimes, then they don't respond other times, then they'll respond, then they won't respond. Sometimes they respond, sometimes they don't. Think of the brain, especially the cortex, as the world's largest orchestra. The neurons are individual players in that orchestra, each playing their own individual instruments. Now the song is playing on, but individual players will play a piece of the song, then they'll be silent, then they'll play again later and they'll be silent. So individual neurons are not very, you can't count on them. They're not very reliable. But nonetheless, the song manages to play on. So there must be some organization at a higher level of individual neurons. Now, Dimitris Panosas, a former postdoc of mine who's now at University College London, who I continue to work with, he published a study last year showing that if you look at the brain's near electric fields, again, these are like local field potentials, but just slightly higher above, just a slight level of summation above local field potentials. He found if you record in the prefrontal cortex and examine the brain's near electric fields, you can actually read the contents of working memory, whatever the picture or item or what the animal's holding in mind in working memory at that moment. Now, we could do that with single neurons long ago, but it requires a lot of averaging across trials because individual neurons are fickle and they're not reliable. So you need to have multiple repetitions before you can start pulling information from the individual neurons. But if you look at these brains' near electric fields, you get a pure information signal that has little or no representational drift. It's a pure signal without all the fickleness of single neurons. So a very reliable signal about what the information the brain is processing. Now that sounds like a useful thing for me, and it's hard for me to imagine evolution would not take advantage of that. Okay, and it isn't just useful for reading information from the brain. It's also useful for producing organization in the brain, right? Because if you want to control what the brain does, you don't want to control the individual players, individual neurons who are fickle. That's hopelessly complex. But if you control things at the level of the song, at this higher level, you can then move around millions of neurons without worrying about this individual fickleness. And this idea that these electric fields are having an influence is not fanciful. It's not far-fetched. It's actually, there's a lot of information, a lot of data, thousands of papers showing examples of effective coupling in the brain, that electric fields have a causal influence on neural activity. Now, there's lots and lots of papers on this. You can go read the literature. Here's a paper we just published this year from the prefrontal cortex showing that during a working memory task, you can actually measure a causative effect of these electric fields on the underlying neural activity. And so the point of this is that there's a lot of communication that's happening in the brain that's outside of synapses and spikes. I'm not saying synapses and spikes aren't important. They're foundational. Everything begins with them, but there's more going on in the brain than just synaptic transmission generated by spiking activity. And for further illustration of that, consider this EM scan of cortical neurons. And what you can see is their cell bodies are very close together. Their cell bodies are clustered together, right? Well, there's no synapses at the cell bodies. All the synapses are taking place out at the dendrites and the axons, which surround these cell bodies. But these cell bodies are smushing themselves together, pressing themselves together. Why is that? Because these are large expanses of unmyelinated membrane that's perfect for a direct electrical influence between neurons. And Demetrius and I took this even further. Demetrius mainly actually has taken this even further by postulating something called cytoelectric coupling. That electric fields not only influence the brain's activity and change how the brain communicates, regulate how the brain communicates, they can actually, these fluctuating electric fields can actually restructure the brain on the molecular level, on the protein level that builds networks, allowing networks to fine-tune themselves to process information more efficiently. The electric fields carry information. And the electric fields can actually fine-tune the structure of the brain at the molecular level. They make the brain process information more efficiently. And to get an idea what this is about, some people may recognize this as if you put grains of sand on a speaker and start playing notes, the grains of sand will organize themselves based on the sound waves. And that's pretty much what we think is happening at the level of the structural proteins in the brain by these fluctuating electric fields. It's a way for the brain's information to influence the brain's infrastructure down at the very detailed level. So the way we think about this is that brain anatomy is like the road and highway system. The road and highway system is very important. Traffic doesn't go anywhere unless you have a road and highway system, but it only says where traffic could go. Where traffic does go, what your thoughts are from moment to moment is how the traffic actually flows on your anatomy from moment to moment, how the traffic actually flows on this road and highway system. And we think these patterns of oscillatory dynamics, these emergent properties, are what directs the traffic. Now, is there any evidence for this? Well, yes. When Tim Bushman was in my laboratory, now a professor at Princeton University, he studied monkeys as they were switching back and forth between two different task rules. One was match and non-match. Rule 1 was match, rule 2 was non-match. Pick the thing that's identical to what you saw before versus pick the thing that's different. Those are the two rules. And what this shows is a drawing of the lateral prefrontal cortex. This is this be right here on the side of my head. This is posteriors to the left, anteriors to the right. This is the principal sulcus, arcuate sulcus here, and each of the circles is a recording site where we had an electrode. And what the colored line shows is where we saw increases in synchrony between the local field potentials between two recording sites whenever the animal was following one rule versus the other rule. And as you can see, there's two different patterns of synchrony for the two rules. There's one pattern of LFP synchrony when the animal is following one rule, and then the overlapping but different pattern of synchrony between the recording sites and the LFPs when the animal is following the other rule. Now, we looked across all frequencies from 1 hertz to 100 hertz looking for this effect, and we found it primarily in the beta range, about 12 to 25 hertz. This top-down information, the rules the animal was using to solve the task, we found that the beta synchrony, 12 to 25 hertz LFP synchrony, seemed to form ensembles that represented the task rules. And since Tim did this study, we've done a wide variety of additional studies. We reanalyzed old data, we did new experiments, and every time we've taught the animal some new top-down information, whether it's a category or a rule, categories are like rules anyway. Whenever we taught the animal top-down information, we find the same effect, not only in the prefrontal cortex, but in the parietal cortex, between the prefrontal and parietal cortex, between the prefrontal cortex and the striatum. We'd actually see these patterns of synchrony-based ensembles develop as the animal's learning the information. Okay, so with that in mind, let's turn to what these beta rhythms may do. Michael Lundquist, when he was in the laboratory, he studied the role of beta rhythms in the control of working memory. Now, in this particular task, it was a simple working memory task. The animal saw two pictures in a row, S1 and S2. There was a short memory delay. After the delay, the animal had to tell us what pictures did you just see. Okay, and what Michael did was unusual. Over the history of neurophysiology, what we generally do is we record electrical activity across identical conditions. There might be 20 trials of the same condition, 20 trials of another condition, so on. Then we average across identical trials to try to boost the signal and decrease the noise. But Michael thought, well, maybe this noise, so-called noise that we're seeing trial, maybe it isn't noise, maybe it's actually real activity. It's a real variation, real important what the brain does. So what Michael did is he studied neural activity in real time without averaging. And this is what he found. So here's an example of just a single trial from the prefrontal cortex during this working memory task. Now we're looking at local field potentials. This is time on the x-axis. So here's when the animal saw the first picture, S1. There was a short gap in time. Second picture, S2. Then there was a longer gap in time. And out here at the end of the trial, the monkey told us what pictures it saw. What's shown on the y-axis is LFP frequency. We're plotting here from 0 to 120 hertz. And here's the power at each frequency. So what did we find? We didn't find this. Much of what we thought about working memory usually involved just elevated, sustained activity. That's because of all this trial averaging, we thought that working memory activity was just simply keeping neurons activated while the animal's holding something in memory. But we now see, looking at this real time, that it's not continuous sustained activity. It's this very bursty kind of complex set of dynamics. And what we found is that the stimulus information about what pictures the monkey was holding in mind, they were carried by spiking that occurred during bursts of LFP gamma frequencies. Like you see here, here's a burst of a high gamma power, in this case 90 hertz power, 100 hertz power, et cetera, and so forth. Whenever the spiking carrying information was co-occurring both in time and space with these bursts of gamma. The spikes are riding on top of these bursts of gamma. Okay? And what was curious we found was that these bursts of gamma with the spiking carrying the information, the contents of working memory, they alternated with bursts of lower frequency beta, shown here in red. And in fact, we found that beta and gamma were actually mirror images of one another. They were anti-correlated. So now we're looking at the average across trials, and red's the gamma burst rate and brown's the beta burst rate. The burst rate of the gamma, remember the spiking is associated with this gamma bursting, and you see the gamma bursting does exactly what we've seen for many years when you look at spiking activity. It goes up during stimulus presentation, then goes back down, and over the memory delay it builds up, which is exactly what you see for spiking if you ever record spiking in the cortex during a working memory task. So the spiking and the gamma are intimately related to one another. But curiously enough, beta is doing the opposite. Whenever gamma goes up, beta goes down. Whenever beta goes up, gamma goes down. So beta and gamma are anti-correlated. Well, if gamma and spiking is carrying the contents of working memory, what's beta doing? Well, what beta is doing is carrying top-down information. It's carrying the rules the animal needs to solve the task. The gamma and spiking is carrying the contents of working memory. The beta is carrying the rules the animal is using to solve the task, how to do the working memory task. So the idea here is that beta gates gamma. Beta is an inhibitory signal that controls the expression of gamma, and that's how the brain controls access to working memory, how the brain regulates what gets into working memory, what gets held into working memory. The idea is that beta patterns are top-down inhibitory control signals. And what this beta does, it reduces gamma and spiking related to bottom-up sensory information wherever it lands in cortex. Okay, so beta is playing a gatekeeper role in determining where information can be expressed. In this case, what information gets into working memory and what information can be cleared out of working memory. The idea is that, and we recently distilled all this work into a publication just published earlier this year, where we propose a new theory called spatial computing. And the idea is these kind of beta gamma dynamics do something. They allow the brain to control its own process. They allow this top-down executive control. The idea is the brain applies beta rhythms to physical patches of cortex to selectively control just the right neurons at the right time to do the right things. Beta is the orchestra conductor I referred to earlier. Now, this is all nice words. So the idea is to think that beta is kind of like a stencil. Beta is inhibitory, so if you want activity to appear somewhere in the cortex, you put beta other places, and beta, gamma, and spiking can be expressed wherever beta is not. So beta is like a stencil. So let me unpack this a bit to make this a little clearer. Here, imagine this, here's spatial computing explained. Now imagine a task where we have the animal remember 2 pictures, a working memory task. The animal has to remember 2 pictures and their order of appearance. So if I show the animal padlock and then butterfly, the animal has to respond padlock and butterfly. If I show a butterfly and padlock, it has to respond butterfly then padlock. And it could be other pictures like house and butterfly, or umbrella and padlock. That's the task. And if you record in prefrontal cortex, you'll find mixed selectivity neurons. In fact, this observation, this exact observation, is what led to the original mixed selectivity paper. What we found when we recorded in the prefrontal cortex during this kind of task is you had neurons that show nonlinear activity to conjunctions of factors. So you might find a neuron in prefrontal cortex that activates to the padlock, but only when the padlock appears first. Or another neuron may only respond to the padlock when it appears second. And you'll find other neurons that will respond only to the butterfly when it's second, or only to the butterfly it's first. If you have 3 or 4 objects in 2 positions, you'll find neurons that will respond to every single combination of object and order, picture and order in the sequence that you could find. So how does this work? Well, here's this is the same patch of cortex, and here's bottom-up information, random connectivity from the padlock and the butterfly, all going up to the same patch of cortex. Again, this is the same patch of cortex here. So you'll find a bunch of neurons that will be activated by the padlock, interspersed with a bunch of neurons that are activated by the butterfly. There'll be some clustering as there is in cortex. The butterfly neurons will be closer to one another, and the padlock neurons will be closer to one another. But generally across cortex, you'll find intermixing, especially higher level cortex. You'll find intermixing of these neurons sensitive to both the padlock and the butterfly. So how does spatial computing work? Spatial computing provides a top-down signal that lets the brain keep track of the rules of the task. And the rule of the task is this object is first, this object is second. That's a rule. Remember the first object and the second object. So it does, beta comes out. Beta has a particular pattern, a template, a stencil that corresponds to first slot. And this beta signal comes down and basically inhibits, this is very simplified of course, beta comes down and actually inhibits half of this patch of cortex. So not only this half of cortex can respond to the first object, the padlock. So now you'll get neurons that only respond to the padlock when the padlock is first, because this beta signal suppresses other responses. And then there's another pattern of beta that corresponds to second slot, second position, and now that deactivates or inhibits another patch of cortex. Now you have neurons here that will only respond to the combination of the butterfly and it being second, because this is where this information is allowed to be expressed, because beta is suppressing everywhere else. So now you have, this is how spatial computing, how these beta patterns explains mixed selectivity. You have neurons that respond to a conjunction of the stimulus and the top-down information by virtue of this inhibitory beta effect. Now the top-down effect will be the same. The pattern for first versus second in beta will always be the same. But you can plug different objects, different pictures into it. So if I present the pictures in the opposite order, you'll find neurons on this patch of cortex that will only respond to the butterfly when it's first, or neurons in this patch of cortex that will only respond to the lock when it's second. And we think this is how the brain controls its own processing. So now in the beginning of the talk, I mentioned that 22 years ago, Jonathan Cohen and I presented this integrative theory of prefrontal cortex function, where we think the prefrontal cortex acts like an orchestra conductor that tells the rest of the brain what to do. Well, back then, 22 years ago, we thought this is a spiking-based model. This all takes place by anatomical connections and spiking influencing neurons, spiking from the prefrontal cortex, influencing neurons back in sensory cortex. But I think of spatial computing as the 21st century update to this. We no longer think that this influence of the prefrontal cortex on the rest of the cortex is based on spiking and synapses alone. It's happening through these oscillatory dynamics. Okay, now when Andre Bastos and Roman Lunis, Andre Bastos is now a professor at Vanderbilt, Roman Lunis is now a medical doctor. When they were in the laboratory, they wanted to examine these beta gamma dynamics in a little more detail. So have recording surfaces along the entire shaft. And what you could do is you can record from all layers of cortex simultaneously. Now, why the interest in different layers of cortex? Well, first of all, cortex has layers, so it must be doing something important. But important for this particular study is that the superficial layers of cortex, everything above layer four and above, are the anatomically are the layers that feed forward sensory information from the back of the brain to the front of the brain, from sensory cortex to executive frontal cortex. That's superficial layers. The deep layers are the ones that feed back information in the opposite direction, from the executive frontal cortex back to sensory cortex. So if this hypothesis that that gamma is important for bottom-up sensory information and beta is important for top-down feedback, well, we should see more gamma in superficial layers and more beta in deep layers of cortex, and they should have an interactive relationship. And that's exactly what they found, summarized in this slide. What they found through these recordings and doing a whole bunch of causality analysis and whatnot, and modeling, they found that top-down information, control signals, are carried by deep layer beta in deep layers of cortex. Deep layer beta regulates weaker superficial layer beta. And the beta and gamma are in both superficial layers. It's that the deep layers have stronger beta and the superficial layers have stronger gamma. Higher gamma. So deep layer beta carrying top-down information regulates the expression of superficial layer beta, and that in turn, through this anti-correlation push-pull relationship, regulates the expression of gamma and spiking carrying bottom-up information, carrying the contents of thought. Okay, now we've conducted this study and devised this theory based on working memory in the prefrontal cortex, but then we thought, why should this be only true for prefrontal cortex alone? If you look at the literature, you'll see these kind of beta-gamma dynamics all over cortex. In fact, the most famous observations from motor cortex, where it was first observed. So we broadened up these studies, and what we found, these beta-gamma laminar dynamics are literally everywhere in cortex. So Diego Mendoza-Halliday from Bob Desmond's lab, along with Alex Major from my laboratory, they put together this beautiful study. They amassed data from a variety of different studies, and they looked at laminar recordings from 14 cortical areas in the primate brain. So I say 14 cortical areas, as much as all of cortex. And what they found, these beta-gamma laminar dynamics were everywhere they looked, everywhere in cortex. And in fact, they were so reliable. You can tell where the middle layer, the input layer, layer 4 of cortex is. You can tell where that is just by knowing where the beta and gamma signals cross over. You can reliably tell where it is everywhere in cortex. And when you see that kind of robustness and generality, you know that this suggests that these beta-gamma laminar dynamics are indeed very important and play a general role in cortical function. In a more recent study, we moved beyond working memory and found these same beta-gamma dynamics also works for predictive coding, your brain's automatic anticipation of what you're about to see, so it can suppress things that are not useful. Okay, so this beta-gamma dynamics play a general role, we think, in virtually everything the cortex does. Now, another series of studies related to these emergent properties is I've been conducting some studies with my colleague Emery Brown at MIT, who's also an anesthesiologist. And we're asking the question, how does general anesthesia cause loss of consciousness? This is a nice collaboration because Emery liked the joke, I'm interested in consciousness, he's interested in unconsciousness. And this may be disturbing to know, because anesthesia has been around for a very long time, over 100 years, that medical doctors don't really know why general anesthesia causes loss of consciousness. They just know that it works. And there's been this tacit assumption that it just sort of shuts off your cortex. Well, what it doesn't shut off your cortex, we found. What it actually does is it changes these emergent properties, changes these oscillatory dynamics to make them very different from the awake state. So here's a normal 25 seconds of time, recorded from a monkey while the monkey was awake, and these are four different cortical areas: frontal eye fields, prefrontal cortex, so frontal cortex. This is parietal cortex, and this is auditory cortex, and these are LFPs recorded in real time. And here's spiking activity from the same electrodes in real time. And what you see is a lot of high frequency chatter because all these beta gamma things are happening constantly. Ensembles are forming and they're breaking. It's all happening at these higher frequency levels. So you see a lot of high frequency radio chatter in the cortex. But then we anesthetize the animal with a common anesthetic called propofol. And we find that all the high frequencies we just associated with cognition are all replaced by this low frequency delta 1 hertz hum. Okay, so what anesthesia doesn't just shut off your cortex, it replaces all the high frequencies we associate with cognition with this low frequency hum that's incompatible with cognition. And to test that hypothesis, we actually tried to restore high frequency oscillatory dynamics to the cortex by high frequency electrical stimulation of the anterior thalamic nuclei. We thought if we can hit the thalamic nuclei with high frequencies, we might be able to restore this awake state to the animal's cortex. The animals would wake up. And that's exactly what happened. The animals are fully anesthetized, and by stimulating the thalamus, the cortex returned to this high frequency FM radio chatter state, and the animals woke up from anesthesia. Then we turned off electrical stimulation. They stayed awake for a few seconds, then went back to sleep again. Okay. Now, these oscillatory dynamics, I've been talking about them as if they're like, broadcast signals that tell the cortex do this or do that, but they're much more nuanced and complex than that. These oscillatory dynamics don't just they're not standing waves. They're not like a jump rope where you're just going up and down in place. They actually move across cortex. They form traveling waves that move across cortex. And now the oscillatories are forming these waves that move. Now you can have very precise control over different networks in cortex by how and where the waves move. So here's an example of a recording. This is a raw recording of electric voltages from the prefrontal cortex. This is an electrode array. Each square is a different electrode. And it's recorded in real time as animals solving a working memory task. And what you get, this particular wave here is a beta wave around 20 hertz. And it's hard to see from this real-time plot, but what's going on is the beta wave isn't just standing in place, it's rotating around the cortex, actually rotating around the cortex. And sure enough, here's now a summation from the same subject. Here's the lateral prefrontal cortex, and this time this is posterior on the right, anterior on the left. Here's the principal sulcus, so right here on the side of my head. And what you see is these waves, these traveling waves, they don't just bounce around the cortex randomly like ping pong balls or billiard balls hitting one another. They move in very specific ways, just like the crowd doing the wave. They move in very stereotyped fashion. So in this case, the animal's solving this task, and the waves moved in two directions, from dorsal to posterior, and from anterior to dorsal to ventral posterior, and in the opposite direction, from ventral to dorsal and posterior. So these waves are moving back and forth in very stereotyped ways, very organized, while the monkey's solving this task. And when you see this kind of organization in the brain, it tells you it must be important to function, because everywhere in the brain you see that kind of organization, there's something functionally important going on. And sure enough, these beta waves, they actually change direction of rotation during the working memory task. So for this particular subject, when the animal is waiting to start the task, the waves are kind of moving in both directions back and forth. Then once the animal starts the task, one direction drops out and the waves just start moving in just one direction only while the animal's solving the task. Then here at the end of the trial, when the animal gives its answer, then the waves go back to what they were doing before the task started. So I should mention this is a time on the x-axis, and this is again the stimulus being presented, memory delay. This is baseline when the animal's waiting, and here's after the trial. So the waves are strongly modulated, the direction of the traveling of waves are strongly modulated by the cognitive demands of the task. In general, traveling waves are really super useful. They buffer information about time and recent network activity. They just by their existence, they do things that are very crucial to neural computation. Now, the way to explain this is if I took a video of a pond and I dropped 4 pebbles in the pond one at a time, I could show you a still photograph of the video like I'm showing you here, and you could tell me where the pebbles dropped, how long ago they dropped. Just by virtue of the fact that the waves are moving in a stereotype fashion. And this is what traveling waves do for your brain. Just the fact that they're moving across cortex, they automatically maintain information about recent network history just from the pattern of waves alone, which is crucial for neural computation. Finally, one question this brings up, all this complex waves and this sparse activity brings up a question of how the brain maintains information between these waves of activity. Well, we saw the traveling waves are useful for buffering information. There's another way that the brain can hold on to information in between bouts of waves and spiking activity, and that is short-term synaptic plasticity. Computational modeling suggests that short-term synaptic plasticity exists in cortex. What happens is whenever a spiking happens, it potentiates synapses for about 750 milliseconds, about 1 second, based on calcium dynamics. This is one of the last papers of Pat Goldman-Rakesh, by the way. And what that means is that whenever the network spikes, there's an after image, an impression in the network that carries the information between spikes. So Leo Kosakov and Adam Eisen, graduate students in my laboratory, they built a model that had a working memory that used the short-term synaptic plasticity. And it worked. Well, of course it worked, so they built the model to have short-term synaptic plasticity, so of course it's going to work. But what we're interested in is what else happens to the model? What else can the model do that we didn't bake into the model, we didn't build into the model? And what they found is that just having short-term synaptic plasticity in this model made the neural networks function better in ways that we didn't build into the model. So for example, it makes neural networks more robust to noise. You take a input signal like a picture and you make it all noisy and stuff. A network without short-term synaptic plasticity has trouble performing when there is any noise into the signal. But with synaptic plasticity, it's extremely robust to noise. It can much like your brain can, you can detect signals even if they're embedded in a little bit of noise. And it's more robust to degradation of synapses. This is called, this idea of a robust degradation is a standard way in which we test the robustness, effectiveness of neural network models. Real networks in the brain need to be robust to degradation because you're constantly losing neurons, losing synapses. Well, they found that a model of working memory without short-term synaptic plasticity, one based on just sustained spiking alone, that model is not very robust. If you delete as little as 10% of the synapses, the model falls apart and can't solve the task. But in a model with short-term synaptic plasticity, you can take away as many as 40% of the synapses and the model still performs just fine. So that's a graceful degradation, and that's a key component that your brain needs to do it. And the fact that these networks do it with this small addition tells you that short-term synaptic plasticity must play an important role in network function. And finally, it helps stabilize networks so that neural activity doesn't run wild. A bunch of excitatory units all connected together in networks will tend to go into epileptic form activity, tend to run wild as positive feedback loops reinforce one another. But with the short-term synaptic plasticity and the spare spiking, the network stays stable and keeps doing reliable computations over and over again without this runaway activity. So with that, I'll sum up. And the take-home messages from my talk are the neural activity underlying cognition is not smooth or steady state. It's complex, dynamic, and rhythmic. There are emergent properties like traveling waves that are providing new insights into mechanisms of function that we didn't even dream about back in the 20th century. More specifically, lower frequency beta, sometimes it dips into alpha, and alpha is right below beta, around 10 hertz or so. So these distinctions in different frequency bands, alpha, beta, they're arbitrary distinctions. They were made up over 100 years ago, so we can't be too strict on, they're not sharp dividing lines. But these lower frequency waves are the control signals. They control the higher frequency gamma waves that in turn are co-localized with spiking that carry the contents of thought. And that way, top-down controls bottom-up, which is what your fundamental problem your brain needs to solve. You need to be able to control what your brain does. And we think this is a cortical motif that plays a general role in regulating all cortical functions, not just working memory, attention, predictive coding, virtually anything the cortex does. So I thank you for your attention, And I thank all the students in my laboratory and postdocs in my laboratory who actually do all the hard work in conducting these studies. Thank you very much.
Thank you so much, Earl, for this fascinating talk.